Hello, and thank you for joining us today for the 137th episode of the Learning with Lowell show. Today we are joined with Dr. Varun, who is in the trenches in the longevity, aging, and epigenetic spaces. He is the head of Bioinformatics at Truediagnostics, where for the last 2 years he's been working to apply his passion in longevity and science to develop bioaging clocks. In this episode, we learn about DNA methylation. We actually get quite nerdy on that in the first 30 minutes. Biomarkers, how bioaging clocks are developed, why they are so important, and the results of developing really powerfully accurate clocks. We see a lot. We talk about some of the results he's seen. We also discuss the future of longevity space, axolotl genomic nerdiness, research recommendations, and much more. Remember to subscribe, tell your friends to let me know if you like this episode or what you thought about it. Also, we have a new YouTube and clips channel for the video components of these interviews. Check out the show notes to see that. And finally, let's stay curious, learn about Dr. Varun, longevity, and the aging space. I thought it'd be really great just to define what is methylation, just for people who don't know, and then we can start just getting deeper down in the rabbit hole. And what is like your fascination with it? Like, what is the thing that you really find exciting about methylation?
Yeah. So DNA. So specifically, when we think about DNA methylation,
Oh, I
it's
said it wrong.
no, not a problem. It took me a minute to figure that out too. DNA methylation is essentially the addition of a methyl group, that a specific amino acid to the DNA itself, and what that allows is essentially a ability to read the DNA in a different way. So when we start to think about, how DNA gets transcribed into RNA and then translated to proteins, their transcription allows that DNA to go to RNA, and some of that RNA becomes messenger RNA, and then allows that to become proteins, which actually do a lot of the biological process or cultivate a lot of that biological processes. And so DNA methylation exists as a way to essentially, without changing the DNA itself, it reads the DNA, it affects the transcription of the DNA so that it allows for different phenotypes or different biological processes to be generated. And so even though you're not changing the DNA itself, you're essentially allowing it to be read differently, which then can cause changes that permeate throughout the biology of whatever organism that you're studying.
Hmm. How did we discover it? Was it just there waiting to happen, kind of like CRISPR in the bacteria, or is it like how do we figure this out?
It was, see, I forget the name of the individual, but, it was a researcher out in, I think Adrian Bird was one of the uh, first researchers that really figured it out in vertebrates, and also looked at CpG methylation specifically. And essentially, it came from a lot of the X-ray crystallography that was done
Hmm.
before, um, and started to see that a lot of these amino acids were being added onto the DNA itself.
Okay. And so
Yeah,
it's not like a new thing. It sounds like, crystallography, I think that's how we first understood what the DNA helix looked
DNA
like.
itself, yeah.
Yeah. So that's like
Um,
70 years old.
but but the
So
thi
it's
but the thing about it is that, not until really the height of sequencing, and this is the case with DNA, that we really are starting to see, really understand more of it, in terms of understanding, which locations, that it's being, methylated at. Um, it's popularly noted that, DNA methylation specifically occurs at cytosines, and so, really looking at, cytosine methylation, is kind of where we think it is right now. But moving forward, as sequencing, becomes a little bit more, popularized, we might start to see it, occur elsewhere.
is it possible for there to be like other hidden things going on, like methylation that we just don't know about?
I imagine so. So
Okay.
another facet of DNA methylation is one type of epigenetic mark, and so this is
Hmm.
epigenetics. Essentially means that alteration of the genetics without actually changing the genome itself. And so you actually have something called histone modifications, which those methylation marks and other type of amino acids are being added to the histone. So the proteins that really coil the DNA and condense it so that it fits into the cell. And so the reason why I bring that up is because even though we know that CpG methylation or cytosine methylation is usually a methyl group at being added, know that in histones at those on those proteins, not only methyl groups are being added, but acetyl groups and other types of amino acids are being added there. So who knows? Maybe something like that also occurs in the DNA itself, and probably somebody's already figured this out. So I should, staying on top of the scientific literature is a must, especially with recent advances in technology.
Histones. I'm gonna definitely have to look into that deeper because I've been like there's a side interest of mine trying to understand like how prion diseases exist or like how they work so that we can like I'm kind of curious like how people are how we could like combat them in an effective way. So that's another thing that I can start looking into. But you mentioned something that's actually really important, which is like how do you stay up to date on things? And this can tie into like this axolotl story that I know we wanted to get into. But I was
Sure.
reading a study that said that after 18 months outside of university, like your knowledge base is basically it's not the cutting edge anymore. And so I am curious, how do you stay on the cutting edge? Like what do you have to do even on like a weekly basis to stay there?
Honestly, I'm gonna let you in on a little secret. We try to stay on top of it, but it's very difficult. But what I try to do as much as I can is so especially in this day of age, having Google Scholar is
Mm,
especially sending those alerts, it has been it's been ridiculously important for staying on top of everything. Because and on top of that too, I have certain searches just for key terms. For example, when I was a grad student, I had a Google Scholar alert for anything had to do with axolotl regeneration and axolotl genomics as another keyword. But nowadays, since I have been focusing more on epigenetic aging and epigenetic tests and things like that, um, my keywords have really changed in the recent years. And as much as I try to stay on top of the axolotl literature, it really goes within the focus. So
mm,
Google Scholar has been very helpful, and I think with the internet age, I think Google has really helped out in at least amassing that information. Number two is ironically Twitter. And I say ironically because there's all this debate about whether you could believe stuff on Twitter, and not saying that you have to, but if you find that a lot of the researchers that you read, some of them are very active on Twitter, and not only those researchers but other researchers in the field, and so they'll tweet out publications that really made an impact for them, and naturally that kind of provides a resource for me to look back on and go from there. Um,
mm,
but it was difficult even now to because you could read all the modern literature and all the new stuff, but it is very imperative to go back to where it first came from. So, for example, the DNA methylation question is actually I don't even know if I answered it 100%, but you have to kind of understand the history before going into to the modern times. But Adrian Bird was one of them that really a lot of his original work, I think, with CpG identifying CpG islands and CpG methylation really stood out just because of how it directly related over to understanding vertebrate DNA methylation or cytosine methylation.
mhm. And just as like a slight transition, so we could have like this little axolotl story, and then we can because that's more of like a regenerative
Yeah.
uh thing, and then we're gonna like the majority of this episode I think is gonna be on longevity. Um, what is the fascination with axolotls like other than I imagine to get a PhD, but I mean I imagine there's some passion underneath that as well.
Yeah, I mean, uh, how does so when I joined, my fascination with the axolotl was regeneration. I watched the Amazing Spider-Man, I think the one with the Andrew Garfield, the
Mhm,
reboot. And even though the basis of that movie was a lizard, the axolotl had this innate ability to regenerate not only limbs but spinal cord, half the brain, virtually all these muscles inside of it. And so naturally the question then becomes, how does this happen? And so that was really the first part, is maybe unlocking some of those or just identifying some of those programs, so then potentially we could translate that over to humans. So that was step one. But then step two, I think from a bioinformatics perspective and a computational biology perspective, as a trainee, I really wanted to understand how their genome was built, or what makes their genome unique. Because again, as I mentioned, that 32 gigabases, that's a lot of DNA. And how do you fit that into a cell of a of a smaller creature, right? I mean, if you compare the size of an axolotl to a human, and we have 10 times less the size of their DNA, how does that get packaged into such a strong and into such a small body? And so that was another fascination as well. And so providing, being able to uh, essentially study that, but then also provide some resources for the community to maybe take that research a little bit further. That's what excited me about that field. And it really made me, I think it made me a lot more appreciative of the human model, because a lot of the bioinformatics resources, the genome, like a lot of that wasn't built when I first started. And so, you were kind of navigating through the Wild West. And I'd like to think, maybe finding the same problems that that people originally had with, even developing the human genome or human methylome during that time.
Mhm. I think the weirdest thing about the axolotl, like, isn't the fact that they regenerate. I mean, that is weird.
Yeah.
Like they regenerate all these things, but when it's regenerated, it works. It's not just like a hump. Like it's not like a lump of flesh,
Yeah.
or something like it's like some of the like the cell ag stuff going on, or even when they're trying to make like heart tissue or something, they have to do some work before it can do what it was. What the original cells were, but it'll grow an entire hand and it'll just start walking with it. It's like that's weird. That's like
Yeah.
it just comes out. I mean, unless I'm misremembering, but just like it regrows the arm and the arm just starts working. Like they don't have to do like physical training or anything. Like there's just that's such a weird thing, given like all the stuff that we have to do just to maintain. If we break a leg, all the work we have to do just to get that to kind of be realigned. And there's no,
Yeah.
it's
No,
entirely different. Yeah.
I think that's absolutely true. And one thing that really makes it a model system for regeneration is the fact that if you look at their limbs, I mean, they have four fingers, so it does look like this, but the composition of those of that limb is very similar. They have the humerus, they have the sorry, the forelimb bone. They got the digits. They have what is it? All the blood vascular system is very similar and akin to that of the human the human arm, and so it provides at least like some sort of translational um, some sort of translation between the two models. And actually, to your point, like when we start the other model that is typically studied in regeneration is the lizard tail. But the problem with the lizard tail is that it comes back different than than the original tail that was cut off. It's actually a lot more cartilaginous than actually with the spinal cord and everything. And you can actually see with a lot of the there was a paper that came out a while back where they showed that the cell makeup is completely different than when it came before, or when it was before, which is not the case with the axolotl limb. It's exact. It's almost exact.
That's weird. And then like you were mentioning with the bigger genome, like is it all useful in a sense? Like is it all adding up to the conclusion of like a working organism? Or do you think there's stuff in there that's like just kind of like carried over like luggage from the
Uh,
past?
I would actually. So I will preface this by saying since the genome came out, I think back in 2019, and then the chromosomal assembly was 2021. We're still trying to unpack that, but a lot of it is repetitive, quote unquote, junk DNA. But What's interesting is if you look at some of these regenerative organisms, such as planaria, the flatworm, they also are, like, I think, greater than 50% repetitive genome. And so this is where we wonder, yeah, like it might be quote unquote junk or repetitive DNA, but what if, I mean, time will tell, maybe it does serve a purpose in the biological function. So um, I don't know. And I think that's what's exciting is that now with a lot of the sequencing and the genomic world coming to play, um, that's where I think that's where really this next gen sequencing and all that has been helping us understand these biological processes a little bit more.
Does the genome is the genome stagnant your entire life, or does it decrease by any measure? It's like when you're born, you have a genome, and then when you die 80 years later, whatever, is it the same more or less, or does it decrease at all?
Oh man, that's a great question. Um, Not sure. I guess that's something to read about.
Yeah, I was thinking, I was like, what if like the extra like junk DNA is like extra copier ink, so so you can make more, without it degrading like the telomeres and all this other stuff. So you have like more of a backlog to use
Right.
from if there's some like degradation or like so you have more fidelity.
Yeah, I think in that respect it does decrease. I mean, yeah, like as you kind of already kind of hit those poly A tails at the end of well, I mean, I guess that's more RNA. So RNA does degrade in that sense. In the case of the duplication, actually, a lot of it was a genomic duplication that occurred for axolotls. That occurred, I think, I guess a long time back in the entire scope of their in terms of their family separating from the frog. And so that's kind of that could be a reason why their genome is just completely is so large is through that
Mhm.
duplication.
Yeah.
But to answer your question, yeah, very good question.
Yeah. Well, it leads me to like a general thought, which then leads us to like a much smoother transition. But the if the like if you could have a person never have any genetic degradation, like just like we're like perfect copies our entire life, do you think there would be like a cap? Would you think there'd still be like a cap in terms of how long we could live? Assuming like no one like runs us over. Like would there so like, is like true longevity and increasing the human lifespan like endless, or is there like do you think there's like an inherent physical cap even if it is like a perfect specimen that like does not degrade?
That's a fantastic question. I think that, yeah, so a lot of the aging diseases do come with some of this, I guess, through degradation of key DNA. If you didn't have that, ultimately I think the issue there would then become mutations. And so, for example, the longer that you are in this world, you're more prone to a lot of mutations that occur. Maybe and some of those mutations can cause the removal of a stop codon or the introduction of a start codon, which then ultimately could create a more cancerous environment. So, cancer is one of those examples that I imagine that even if we weren't able to even if we had the ability to not degrade the genome or lose any of that key information, you have a higher level of just being able to mutate regions that could be very negatively that could reduce our longevity ability, I guess. And so I think that yeah, reducing the size of the genome or removing that degradation is not the only thing. We'll have to figure out how to, maintain the original form and the function of that original genome as well.
Mhm. Are there concerns that you have as we develop these technologies in terms of limitations that you worry about? Like, so, like, we have expanded our lifespan, but have we expanded our health span? Would be one example of that. Are there other things that you worry about as you're like in there? Um, no, you're in the field, so
Right. No, yeah, that's
you're in the trenches.
good question. I mean, what I think what I worry about the most is how do you I think it's more of the population aspect, right? Like, yeah, you can increase the longevity of individuals, um, but with overpopulation, that could also cause some other, I guess, um, issues with, like civilization. Literally, like, how do you work with the average individual living much longer, and what does that do to the environment? So that's one thing in a weird way that I think about. Second is, if you increase your longevity, yeah, you want to make sure that, as you mentioned, the health of that individual is consistently high, is consistently good as well, uh, throughout that longevity. I would much rather have someone who maybe lived to the age of 80, but then never really had any ailments, versus an individual that lived to 120, but that started having ailments at the age of 50, because that quality of life really, I mean, I wouldn't want to wish that on my enemy, really. So I think that's something that I kind of think about is, as we improve these kind of epigenetic testings, or even just medicine in general, it's about, I don't think it's the question of keeping people alive longer. It's how do you keep them healthy, and allow them to have a high quality of life, and try to allow them to live a little bit longer.
Yeah. It's like when they have life, are they living? So a
Yeah,
guy I know a lot of elder people where it's like they give off they give off the vibe, if not say it, they wish they could die. It's like they don't it's like I don't see my friends. I can't do any things that I enjoy anymore.
yeah,
It's like, what am I doing? It's like I guess I'm here for my family, but at a certain point it's like I can't do anything with them. Like I'm not in the role I used to be as a caregiver or anything. And so it's like they're in like this like stasis, like they're waiting for a new chapter to begin, but like the chapter's the end of the book. It's like
yeah.
I don't know. So um
That's absolutely true. Yeah, yeah.
um so you're a bioinformatician. So you're more of a numbers, machine learning, crunching type person, up like software type person. There's like people who develop like pharmaceuticals and that type of thing, and you would build like the infrastructure for developing pharmaceuticals, for instance, or develop testing that would help better understand if pharmaceuticals working, or to like better measure populations and that type of thing. Is that a good way to describe kind of like how you fit into the bar like the space?
Yeah, yeah. I think the testing aspect is really key. So as a bioinformatician, how this kind of, I think we're comparable to data analysts, but the key difference here is that we also have a significant biology background, and so a lot of the sequencing data and a lot of the data that really, sorry, the biological big data that comes in, we have some technical, or we have the technical tools or and skills to be able to code and have a statistical mindset. But then when we're parsing through some of the data that comes out, the key is how do you discern, like, maybe some, we'll use the term biomarkers that maybe aren't really important, versus biomarkers that are actually truly important based on the biological literature. And how do you differentiate between maybe just artifacts of the data versus maybe something that is actually being brought up by the signal, a biological signal?
Mhm. So like in many ways, like the machine learning, all this other like they're like tools to help you sift through
Right.
the DNA, like to find the gold nuggets, so that you can see like, all right, here's some good gold veins. At least like kind of a weird metaphor,
Yeah.
uh, to like do more work on.
Exactly, and if you think about machine learning, or even just, your linear regression modeling, and your simple, like, even if you take it to a simple statistical modeling, it's a model. It's going to pull out some features, right? Especially if you have a large enough data set. But then how do you know that those features are truly features, but then versus the just artifacts of the data? And so
Mm,
I think that in order to create a very good model, and again, the AI, with artificial intelligence, it's a kind of a black box still. We're trying to figure out, how does this model work? What is it really picking up? And how do we recapitulate it in terms of understanding the biology? So I think bioinformatics really works in multiple of those facets, and but then really trying not to forget the biology, which, as we kind of talked about, it's really hard to stay up to date. So, we try to have a good, um, uh, I guess, mediation between data-driven understanding, and then also kind of, taking what we already know in the field and trying to apply there.
Chi- um, so like machine learning is kind of like a Chinese box, if I remember in the philosophical concept, where it's like a mechanism that has inputs and it doesn't really know, like it's like it could be Chinese and they don't read it, but they just have like the different letters and they sift through and then have the output. So it's not like I guess that's the difference between like AI, where they're like trying to have like a thinking thing that has some cerebral ability to understand like what they're generating and putting out, where machine learning sounds like the Chinese box, where like the person doesn't know the inputs and out. Like this is like a philosophical thing. I'm
Yeah.
not describing this very well. Basically, there's like this a long time ago. A bunch of weirdos sat in a circle and was like, "Hey, what are some ways to think about the world?" And they're like, "You could have a person or an entity, or in this case, like AI or like a thing we build, where it's basically like a box and an input comes in and it has a bunch of squiggles, and they
Yes.
have a tablet that translates those squiggles and." They translate them and put them out, and in this case, there's like an extra level of computation so that they can sift through the vast amount of it and have an output that makes it easier for you to do your work. But I was just thinking like, oh wow, that sounds like a Chinese box. But um, are there in a given week, like what technology are you actually using? Like what is your tech stack? Like what are you actually like working with?
Sure. Yeah. So I do a lot. I'm more trained as a statistician. So for me, I
Hmm.
still do a lot of multivariate linear regression modeling. And so this is your y equals mx plus y equals m1 x1 plus m2 x2, and just kind of a linear regression approach. The machine learning aspect really comes with a lot of my team. We do some deep learning, and so essentially, how kind of going to that metaphor is, we'll train. So the the predictive algorithm that we try to that we've been kind of working on is, given an age, can we identify features or patterns that allow us to predict the biological age if you were given DNA methylation data? So methylation, especially when you look at the data, it can go from 0 to 100% methylation, or 0 to 1 in percentages, or in decimal format, essentially. And which of those features are allowing us? So, like, these cytosine, the CpG sites that I was mentioning, and their methylation levels, which of those CpGs are really associated to that biological age? And can we use that on just data that we don't even know the age of that
Hmm.
individual, but then predict that age? And so
Yeah.
deep learning really helps with, making a model that's, less error prone. So if we think about mean RMSE, so root mean square error, what produces the least amount of error, and also the highest number of or highest association to their actual age versus their predicted age.
So
And
the closer R gets to one, the more predictive it is.
exactly, and we're
Yeah.
looking more at, the R squared value, so as long as R squared is closer to 1, it provides a good model, but then really, so that's one aspect, but, the error rate is actually even, something a little bit we've been kind of focusing on a little bit more, just as a way to say, on a given time, what is the error that we could expect from, one of these algorithms? And so deep
Mhm.
learning provides, from what we've done so far, it seems to provide much less error than a a linear based model, which kind of it makes sense.
So in your role, are you defining the data to look at, like defining like the control variables and all these other things? Or are you taking the data that like the machine learning and like the other members of the team have maybe brought to you, like the data you have on different people, and
Mm.
then doing analysis of that? Like in the sense of like, are you a part of determining what data should be looked at so that you can have a better like like a stronger R1? Like, are you a part of that? Like, I'm kind of curious, like how do you fit into developing
Yeah,
the system?
yeah. So, actually, a lot of when I first started, it was kind of so we had a bunch of DNA methylation data. So the company that I worked for, we have a commercial test where we actually are able to quantify the DNA methylation of those individuals. And so through that, we've actually amassed a ton of data
Mm.
through that. And so when I started, really looking at the data itself and then being able to generate just what we call this an elastic net regression model, which is very similar to a linear regression model, except you can use a resampling to minimize the error of that linear regression. And what that allows us to do then is, this technique has already been utilized in previously, but the really cool thing is that by having all the data that we had about, I think, upwards of 10,000, at that time, it was upwards of 5,000 individuals, all collected in the same lab, which actually is great because then we know exactly what's being done in terms of the collection. And then on top of that, I can always go back to my counterpart in the lab, who ask, hey, some of these samples came out to report very different methylation marks. Was there an issue with sample preparation? And so quality checks are very important, and I take care of a lot of that as well. And so
Interesting.
yeah, so really, I start with the raw data. I do a lot of the quality checks, making sure that they're properly normalized to each other so that you actually can compare apples to apples rather than apples to oranges. And then
Mhm.
if there are any cases of, let's say, batch effects, meaning, you know, a lot of these samples are collected throughout a long time, and so on any given day, maybe the temperature in the lab was a little high, or there was kind of a minuscule error in the machine that caused those readings to be a little bit different. There are statistical methods to actually correct for those, and so I'll do a lot of that QA/QC, and so just so that the output of those normalized batch corrected values are truly uh, essentially cleaned so that
Mhm.
the model doesn't pick up artifacts of that unclean data.
That makes sense.
Yeah.
Sounds really fascinating too, especially because like I have like that I have that type of mind where like it's like a really fun puzzle, like everything's messed up and you're straightening it all out so the
Yeah.
pipelines comes through and you have like this desired output for yourself and the rest of the team. Um, it sounds like it'd be a lot of fun, especially with all the math and stuff you have to do. Is it really complicated? I mean, you mentioned some of the like the regression and other
Yeah.
types of models you use, but like what's the most like in like a kind of a daily basis, like what's the most complicated? Like this is like we're getting far afield from longevity. Let's like
No, 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 that's fine, that's
I'll
fine, yeah.
transition this path, but I'm just curious, like what is the level of what is the how complex is the math you use on a given day? Like is it like Calc 3? Is it like how like where's the level?
So luckily, it's a lot more on the statistics side. I will
Mm,
say, especially with a lot of programs nowadays, like coding languages such as R, I use a lot of R coding languages. These packages are created by themselves, and so I think
mm,
they're already created by other statisticians, like really
okay.
hardcore statisticians. I think the biggest thing is really choosing the right statistical method that fits the data itself. And as nerdy as it sounds, it's really the most underrated thing that you can do is just look at the distribution of your data. And then by looking at the distribution of the data that you have, does it fit a normal distribution? Does it fit
Yeah.
a binomial distribution? And then using that understanding, what is the proper statistical test that allows you to really gain a lot of insight? Because if you use an improper statistical test, you can ultimately be picking out things that actually don't really relate to whatever feature or whatever biological process you're looking at. And so that's where I think that's actually the hardest part is keeping up with just making that decision of, okay, is this the right statistical test or not? And after time, since we work with the same data, it's almost like a rinse and repeat. But sometimes whenever we get new data, then we have to really make that call of, okay, is this fitting the assumptions that are being made for this data type?
Mhm. Yeah, when I was in college, there were I was in like this. I took a graduate level course, and the teacher was talking about how the professor. I don't know what the titles anymore, but
Yeah.
they I don't know. I'm not naming them. So, but they said that the weirdest thing they saw in like the neuroscience field is that everyone always assumed it was a normal distribution.
Yeah.
Like, they just always assumed it was normal, and it's like and I was like, "Oh, that's weird. Why would that matter?" And he's like 30 minutes like, "This is why it matters." Like, you can get false positives, you can have all these
Oh,
different problems pop
absolutely.
up. I guess that gets to the point of, like, why this is really neat because, like, not to create, like, any association between these horrible people I'm about to mention and you and your group, but there are, like, the Elizabeth Holmes out there that'll create, like, false positives, and it's like, you don't even know if you're doing anything worthwhile. You don't, and that has this horrible effect on people when you're trying to develop a test for developing to understand anything. Where now these people are like, oh, now I have a better sense of who I am, like all these different things. So like this type of information, this type of like stats, and like building like a solid foundation is how
Yeah.
you have like something that actually is useful and something that like gets you 20 years in jail. That's the difference. Well, I guess the difference is also, did you know you were doing it? But so you got to really, but uh, like that's just like a big thing that I always worry about, especially when you're talking about anything involving people. It's like when
Yeah.
you have any predictive qualities, or you're looking at something that involves, how long you're going to be here, how good is that life, and you're trying to determine, what's the person's age, essentially, so they can potentially intervene and even affect that, or like see like with that test. I think like kind of I'm jumping the shark a little bit, but with your test that you guys have developed at Truediagnostics, we can then ascertain like what things can you do to intervene on your age to then affect it in a positive way and potentially a negative way as well. You can kind
Yeah.
of do those things, but like all that starts with just a clean base to know like what's positive, what's no false positives and all these types of like type 1 or type 2 errors and all that type of stuff, which is really just I'm kind of like jumping around a little bit in terms of like what I'm describing, but it's like really important stuff. Like
No, you're
it's like
speaking my language. Yeah.
yeah, it's like the bedrock of everything.
And I do wanna add a little bit more to that because, yeah, I that is something that actually keeps me up at night is am I doing this as scientifically accurate as possible? And luckily, within my organization, we truly believe in published research. And
Hmm.
so, for example, we licensed, this one, clock, exclusively licensed, which was published in, eLife, called the Dunedin Pace. And the reason why we kind of go with this, and also a lot of the internal research and development that we do, we want to go through the peer review process because that is one way to at least have some checks and balances to say that are you doing this the proper way. And so and trying to keep things proprietary, I don't think is actually very helpful for a lot of people. And so by publishing this research, we hope that it gives individuals a little bit more comfort that hey, we went through the rigorous peer review process and that we had other people kind of look at it and say, "Are you doing this the right way?" And boy, peer review is can be sometimes difficult. So there's a reason why we go through it.
Hmm. Is there so far, has there been an outcome of these tests that has surprised you? Like as people have used it or as it's been implemented? Like we're definitely like jumping from stats to
Sure.
application. I'm curious. Yeah. Is there something that's either surprised you or that you've been very like pleased at the stress you put on yourself to be accurate?
Yeah. So so one of the things that actually my team was so I personally wasn't involved with, but the Dunedin pace clock that I told you about, or just briefly mentioned, there was a recent preprint that came out, which I has already gone through the peer review process. I'll check one more time, but they used that clock on a trial of caloric restriction. And we've
Mm,
already known that caloric restriction has some positive longevity benefits. But what was interesting to see is that clock actually responded really well and showed a lower aging rate in that in curbing or in caloric restriction relative to just eating as you will. And so this is one of those things that we're trying to move towards is developing these clocks so that they actually respond with trials and interventions. And uh, that clock itself took about they followed a cohort of individuals from 1973 all the way
Mhm.
till the age of 45. And so imagine how long that took to just be able to gather their necessary data and to be able to build that. But for all that time, to be able to see that clock respond really well to caloric restriction relative to other epigenetic clocks, I mean, I can't imagine how the individuals feel. Actually, I do know
Mhm.
because I've talked to them, and they were really happy to see something like that because it really provides a useful tool that is responding well to known interventional trials.
Mhm. It's like one of the biggest fears, and there's so many different philosophies that exist, like the Stoicism or whatever, to help people be comfortable with the fact that we're all going to die one day. Like we all were born, and so far everyone dies at some point, and so it's like it. I imagine it gives some empowerment to know that you can have some intervention, just so that to know that you can have a positive impact on how long you're going to be here with your loved ones and how
Yeah.
um, well you're going to be here at the same time. I'm curious, as you were looking at those, as you're looking at that clock from a statistics standpoint,
Yeah.
what was the biggest jump? You know, in the sense of like, was it like 20s to 30s, 30s, 40s, whatever? Where in terms of like the aging, that like was there a bigger jump in time range for when there was a difference between like one age group and the next, if that makes sense? Like,
Yeah,
or is
yeah,
it
yeah.
all somewhat uniform in the change over time?
So, I can speak about this using our personal cohort of the Truediagnostic users. The individuals that take our blood tests, we actually saw. I kind of ran that clock on a lot of our on our cohort, and what we see is that there is a general increase. That not really too much of a jump, but it's interesting that at the age of 50, the that clock essentially looks at the pace of aging. So it doesn't give you like
Hmm.
a number of 53 or whatever, but on a scale of 0.6 to 1.6, with one being like you're not aging faster or lower, you're aging about the same. It's standardized to
Yeah.
one. What we see is that at age 50, the pace of aging flip flops when you consider the sex of the patient. So pre-50, males age at a slower rate than females, but once 50 hits and then a little bit past, it was interesting to see that the average the aging pace for males is higher and increases that way. So it kind of this is probably the worst place to do it, but kind of it flip flops so that the pace of aging for males is much higher than females, and
Mm.
it continues like that until essentially the end of the age that we actually have looked at.
That's interesting. So then, when you have these types of results, are the principal users of this data other businesses or other like research biopharmaceutical type uh, groups? Like it's more like a B2B, like what your diagnosis does, or is there like a like does the benefit of these clocks being made, does it impact people directly? Like, if that makes sense, are you guys? I
Yeah.
already heard you. I already heard you say that there are people that will take your kit and they'll look at it. But I'm curious in terms of like taking that data and having like something come from it.
Yeah.
Um, does there have to be like a business that applies that data, or is there like consumers that have found a way to apply the data themselves?
So from the business standpoint, I don't I do but from what I hear is
Yeah.
that we have a DTC market, so people can go to our website and get our test. It's approved, and so we'll ship them out the collection kit, and then
Hmm.
they'll get a little bit of the blood. And it's all done at home. We use a tasso method, but there's also like a finger prick kind of method to collect that, and then they ship it back. So there's a DTC market. We also work with a lot of physicians who, cash pay physicians who really work with individuals that are very, very into, seeing, they spend a lot of time on perfecting their their aging and their health and everything, and so they'll get a lot of those tests and incorporate that into, the monthly, if not every 6 month checkup. And so that's another aspect. But we also, and this is where I really where I really spend a lot of my time is work with researchers to see, hey, we have um, we're trying to understand if this specific pharmacological intervention or some type of other intervention, maybe it's just behavioral, how does that affect aging? Because now we actually it's essentially a quantifiable method of saying, are you really um, uh, decreasing your aging rate, or are you actually increasing it? Because before this, it's aging was very subjective, apart from other lab tests that you could take. And so we're able to gather in patients in the blood in samples through all these different routes, and so that allows us to kind of tap into very different individuals and cohorts, if you will. But I will say a lot of people that take our test. I mean, it's not a cheap test. They really care about their health, and so technically there is kind of a healthy cohort bias. But
Hmm.
throughout through what we hope is that by um, developing these tests and also having a more a cheaper offering, we can actually incorporate individuals that maybe are starting their health journey.
Mhm. Makes sense. All right. Um, from like a numbers standpoint, are the clocks modeled onto like the health? Like, can we tell based on the clock like where people are in like a health span point of view as well, or is it strictly like where they are from like an age? I guess they're kind of related. Like you can't like have one without the other. But I'm curious, how are we able to like separate those two things? So like if you were like, let's
Yeah.
say I'm like 40 years old, but then like through your clocks, I've done everything right. I'm at 30 years old. Does it suggest that like as I age, I'll be doing like I'll be healthier as I age? I guess like are we able to see that type of follow through, or like have we done enough research to see that if that'll work out, or is it kind of early stages in terms of seeing how the data's been proven, the test's been proven, but like how it'll play out in the field?
So that's a great and fantastic question. I will so to directly answer that question, what we recommend is to take multiple tests. And this is not a business standpoint, because technically, if you take the test one time and you are, let's say, 10 years younger, let's say instead of being 40, you're reported as 30. That's fantastic. But one of the things that you really can't tell, except there are some buts to it. Um, but what you can't tell is if you're headed towards a pro aging or a quote unquote anti aging or reverse aging phenotype. So that's why if you take two tests, maybe at zero at the time or at one time, and then maybe 6 months later, and you start to see that decrease consistent, or there's a consistent decrease, or just like you're staying the same, that provides a little bit more of a credence of, hey, whatever you're doing right now is either good because you're maintaining that 10-year reduction, or if it's going even lower, it's like, okay, it's actually really improving. um we don't see this much with a lot of actually healthy individuals, because technically you might have maximized your your anti-aging potential, if you will. But individuals that are, maybe not leading this that healthy of a life, we start to see, and they start to really take that back. You start to see massive decreases within that 6 months. So, uh, as much as taking one test is already a good start, I think you really get the power from taking two tests,
Mm,
or multiple time points. With that said, there are different generations of clocks. So the earlier clocks were really trained on those on your chronological age, essentially. We were able to identify CPG sites that really were highly associative and correlated to the chronological age of that sample. But nowadays, what we're starting to do is we're not really focusing more on the chronological age itself, but we're training different types of covariates, such as your lab measures, on top of the DNA, on
mm,
top of, let's say, the time till death, where you can get that from like a Kaplan-Meier estimate, and instead of using chronological age, but using that information to really develop these clocks. And I think some of the most important clocks that I think I can think about are like the Grim age, the Pheno age. And shout outs to Dr. Morgan Levine for PhenoAge, and then Aki Lu and Stephen Horvath for GrimAge. There are a bunch of other authors. I apologize for not listing all of them. And then Dunedin Pace, really, Daniel Belsky, Avashalom Caspi, and Terry Moffat, and a lot of the individuals at Duke. And so I think this has been really paving the way of, again, if you train it to chronological age, you're kind of running into that same issue of you're still basing it off of chronological age, but what we really want to go towards is that biological age. And so I think that now by integrating a lot of those clinical covariates and other types of omic data, not just DNA methylation itself, you can get a much broader representation of your aging potential and aging system as well.
That makes sense. I imagine actuary scientists would be beating you guys, like beating down your door in particular. But I'd be very curious, even though like humans are much more complex than mice and other like primate models that we do a lot of our clinical studies on as we like
Yeah.
transition through the clinical phases. I'd be curious to see, as we learn more about what interventions work, like how much it would we'd be able to intervene and decrease a person's age clock, for lack of a better term, and
Yeah.
like what impact that'd be, because they're like such they have shorter lifespan, so it's easier to see. Like, it takes us 100 years to see if we gain 10. I'm also curious, and you kind of touched on this in terms of where I wanted to go with the question, which is with all the data you have coming in, and you're kind of a data nerd, and you have the DNA methylation, and I
Yeah.
said it right this time. Are there other biomarkers, other partnerships with other integrations that are going on, or other data sets that are existing that you guys already have at Truediagnostics? If you can't tell me, that's fine. But is there some stuff? Is there just some stuff you're really excited about in terms of experimenting with, or gathering the data to find that you've already found some other like correlative stuff that
Yeah.
hasn't been talked about? Like is there like some nerdy, really deep math statistics stuff going on that you've seen that you haven't had a chance to talk about?
No, I think the biggest one that really comes to mind is so we are partnering with some bright individuals at Harvard. We already have partnered, sorry, at Harvard University, and specifically at the Brigham's Women's Hospital. And then on top of that, Seer Inc. And so the reason why I bring these guys up is because what we wanted to do is kind of follow in that in the footsteps of creating a biological clock, but really taking it to the next level. Because nowadays, with a lot of the new sequencing platforms, we are not biased to only a certain number of proteins that we know are associated with age. We're not biased to only a certain number of metabolites. I think the coolest part is now that there's unbiased targeting of, let's say, proteomics, the Seer platform has been very crucial and very pivotal in unbiasedly understanding the level of a specific protein, and then using that as a way to identify maybe other proteins that are highly associated to biological age, without really limiting yourself to let's say your CRPs or IL-6s that traditionally have been really tested. Not saying that they're not important, but what if there's more
Yeah.
that we're not really touching on? And so that's what we've been doing, and we've already collected. So using the biobank at Mass General Brigham's, we've been able to sequence we've been able to let's say quantify the DNA methylation of 1,500 individuals, and the same individuals had DNA methylation characterized, their proteomics using the Seer platform characterized, their metabolomics characterized, and then on top of that, we have all of their like full physiological, tests, like their actual lab tests and their relative, cell levels and things like that, spirometry levels. And so by having all of those matched data sets, we can then hopefully generate a model that takes every single one of those aspects together to give a more complete picture of their aging process.
That sounds like a lifetime of work with all that data and all that all those many different things you could be looking at. How? Yeah, it sounds like a lifetime of work, and it's something that you'll be spending a long time on.
We have to
Yeah,
underline that there are a lot of very bright individuals working on it, and I'm just really happy that Truediagnostic and our team, we're really putting in a lot of energy into there. So, we're part of the cog, but this is stuff that would needs to be done, and
yeah.
we're hoping is that it makes a huge impact in at least um, allowing people to really better understand their aging process.
There's a like a video where I don't know, there's a website where you can start at like a human scale and you can go up to the point where like everything's the size of a human. So like then the sun's the size of a human, then the galaxy's the size of a human, like everything gets bigger, bigger, bigger. But then you can also go down into a person where it starts
Yeah.
like a human, and then there's like the cell the size of a human, and there's stuff inside the cell and it goes down and down, down. And so like biology is so complicated that you really do need all these different partnerships just just to like have any semblance of a hope and understanding it. I think like we're as complicated as some of our most complicated things that we've developed from machine learning or anything else going on. Yet we're so not understood as well. I think the most understood animal on the planet is probably the mice, given all that we do to them.
Yeah, yeah. It's great
We're
to be a
like,
mouse or
yeah,
mouse.
like we've cured I think every different type of cancer for mice. Like they're just waiting their turn. It's like the have you ever seen Hitchhiker's Guide to the Galaxy? But those the two mice that are like actually in control of the entire thing. Like
I
they,
haven't
they set
watched
it
it,
up
but
this way.
I know that scene. Yeah.
Yeah, yeah. Basically, the mice have set it up this way. We work for them. It's not the other way around. Um, how, how do you see I guess it's kind of the same. Do you see yourself progression into this field in the longevity space, or do you see with all they have access to, like you have such I imagine such a buffet of
Yeah.
opportunity in terms of like what you can learn, what you can try out and do. Where do you see yourself going in the next couple of years?
I that's a question that honestly I don't have the answer to. I think that, but what I can say is this. I think if you limit yourself, and this was actually said to me by a professor a while ago, who I deeply respect. Um, if you limit yourself to one type of study, it really limits you to the breadth of biology. Because I think you really hit that point really, really well. I I can't do all this on my own, nor do I have the capacity to. But what I do have are a particular set of skills. Since we're talking about movie references, but all jokes aside, um, it really is like how do you collaborate with individuals, and it's through those collaborations I think that you learn a lot. And as a scientist, you are always a student. You gain some sort of mastery with certain technical skills, but if you consider yourself a master, personally, I think that's kind of that's where you're really failing as a scientist. I think remaining a student and trying to learn as much as you can, and then trying to associate yourself and also collaborate with other individuals, and also bring something to the table. I think that allows you to do really well. And so it's with that kind of frame of mind that if right now aging is really a question that I haven't answered for myself. I don't know, what is really causing aging. Well, how do you really slow down aging? Can you slow it down? I mean, there are examples of how you can slow it down, but you know, how do you develop those tests? This is the hardest question that I'm trying to find the answer to.
Hmm. Makes sense. It's a lifetime of questions. I
Yeah,
think also for I think some like scientists are like you're trained to be humble, but then like if you don't say you're great at something, then people are like, oh, why are we listening to this person? And it's
yeah,
like
yeah,
I think like a real scientist, in my opinion. Please don't burn my house down. But like I imagine like a scientist is just proven how wrong, like how much they don't know every day. And like
yeah.
there's like a great Neil deGrasse Tyson quote where it's like the more you know, the more the outline, the surface area of your ignorance expands.
Right.
And
Yeah.
so uh like the more cutting edge you are, the more you have to feel like just like, man, I don't know anything. Yeah, sorry, go ahead.
Oh, no, I definitely agree with that, and, I think that it's sometimes it's difficult to say, "I don't know," but I think that's where the beauty is that when you say, "I don't know," the difference is that I wanna know, and so you'll spend a little bit extra time, trying to educate yourself on that. I
Mhm.
mean, as I like as you kind of pointed out, I came from axolotls into human aging, and that jump was not an easy one. And so every day I'm still learning and, trying to take this further. But, there's some aspects of like, oh, the axolotl does this, maybe like, is that the thing that we can bring to this table? And so that diversity of thought can be very useful.
Yeah, I think sometimes people feel like they need to know they need to have a map of where they're going. But especially in science, you're not really like you don't have like cartographers where you're at. Like there's like some semblance that like, hey, we might be in St. Louis, and
Yeah,
I think there might be an ocean if we keep heading west. Um, but like a lot of it's unknown,
yeah,
and surrounding yourself with people that are also that way is like really exciting.
yeah.
Like having like in any industry, like when you're in a team where like not everyone's like, no, I know what's going on. They start there's like a organization called Toastmasters where it's like like a bunch of leaders or something like people who want to be leaders like learn how to speak well
Yeah.
or speak good if that's how I'd say it. But like it's basically like who can speak up the loudest. So it's like I went to their meeting. I went to a meeting and I just like walked out in 5 minutes. I was like, I will lead us out of here. That's it. So like it's like the humbleness is good, especially when you have a team of people that it like they can be like investor facing where it's like, oh yeah, we're great
Right.
at this, but like amongst each other, it's like, man, what is this? So I am curious, what are there elements of what you're working on right now that you're particularly curious about, or in the longevity space that you're
Mm.
particularly curious about right now?
Yeah, so I think, like, for example, there's one study that we're working on, which has kind of opened up my eyes to, maybe reevaluating some of these previous, reevaluating or evaluating from some of these interventions. Um, so the one study that I'm working on is the effect of senolytics, and so how like does clearing out senescent cells provide a more anti-aging, I guess phenotype, if you will. And so it's been reported that, you see a lot of clearance of those senolytic cells, those essentially cells that have pretty much those senescent cells that are being destroyed. And so we wanted to see how these clocks kind of respond to that. And while the study is still undergoing, we see a variation in those clocks, where some of the clocks are reporting that, that there is a decrease in aging, where some of them say there's no response to this one senolytic drug called dasatinib quercetin. After the application of dasatinib quercetin, we see that the epigenetic clocks are not responding uniformly. And so, developing a clock that, again, it kind of goes back to, the research and development side. So developing a clock that's sensitive to that, that's awesome. If it was able to look at those mediations. Number two is exercise. We know that exercise improves the overall aging experience by essentially decreasing the aging, I guess, ailments, if you will. But, is there a difference in different types of exercise? And so, I think that's an area that we're currently working on with, Auburn University. And that, I mean, I try to stay healthy, and I've been, in a I try to do as much exercise as I can. Um, and so, having something, how do what are these clocks telling us about that? It does exercise help. And, I mean, we're still waiting on some of those results back, but, we know that, cardio is incredibly important, for, maintaining a good cardiovascular system that translate to an overall systemic, anti-aging effect, or
Mhm.
reduced, or I guess improved aging effect.
I
Eh,
think that the nicest. Sometimes people feel like they're not healthy now. Like, oh, it's already too late. But I think one of the really interesting things that we've had with these bio clocks, aging clocks. I'm just gonna keep inventing new ways to describe what you've already described. But
Sure.
uh, these aging clocks is like you're showing that at multiple different times can you have an effect on yourself. And with there's lots of studies out there where it comes to like you could be 60 years old and you start getting fit, and it has a huge impact on your brain, it has a huge impact on your heart. Like the human body is so adaptable.
Yes.
It's weird. It's weird how adaptable we are and yet how weak. You punch someone the right way and like lights out. But like, uh it's weird. Organisms really just want to live, but like it makes sense, like you say that. But like it, so like if you're out there and you're thinking like, oh, I'm not healthy, or I feel like I'm not healthy. I see all these things and it just says I'm in a bad spot. Like there's so much you can do, and that's really cool about this research. Is
Yeah.
sometimes it's like if I put in all this work, will I have the outcome? And it's like we're finding more research and more tests that'll show that if you do this work, you will have that outcome. And I think that is really helpful for people who maybe never had that encouragement. And sometimes data is the best encouragement you can have because it's like numbers can be misleading, but to some extent they cannot lie.
Yeah, and I mean, the proof is in the pudding, or proof is in the data, really.
Hmm.
And that's the thing. It's like, I am trying. Uh, constantly improving myself to be a better neuroscientist or an aging scientist, by using data as a way to kind of learn a little bit more. Because by having this tool, now we can objectively and quantifiably say if something is, if if a treatment or a behavioral change or a lifestyle is truly uh, allowing for a better aging environment. And so, just by doing it I've always taken a data-driven approach in learning, and using that as a way to say, "Okay, now go back to the literature, and what does that say?" So, um, some would argue it's not really the best way to learn, but I also don't think that there is a one way to learn. There's different methods of doing it.
Yeah, I think there's a probably a core framework for learning anything, but I'm
Yeah.
not smart enough to know it. But if you look at it, like everyone learns roughly a similar way, because the brain works roughly a similar way for everybody. So whatever works for you. I
Yeah.
mean, you're doing great work. I mean, that's kind of also like one of those things. Like, am I learning in the right way? Well, I'm building things that have never been built before. Like I'm doing math and stats that have never, that are having this type of impact. That kind of like shows you're in the right direction in terms of like you're learning. It's like at this point in your life, if you were learning wrong, I feel like someone would have told
Someone
you.
would have, yeah, someone would have said so, yeah.
Yeah, in your
I mean,
PhD program, all these different
yeah,
things you're doing.
but some people can say that, but, that you're learning a wrong way, but, sometimes you can prove them wrong.
Yeah. So you're I think you're about 2 years in at Truediagnostics.
Yes.
How has that impacted your life? Do you like go around and just tell people about longevity all the time? Are you, as you're learning these new outcomes for your work? Like, I'm just curious, like how does being immersed in this field affect your personal life?
Oh
Are
my
you able to
god.
are you able to firewall it? I don't know.
Yeah, so it's a mix of both almost, because I truly am passionate about this information. But I think one thing, like, of everything I learn and, what I'm doing at work, and I think it was kind of similar during my grad school, because you are really investing a lot of your time and mental efforts, and just, life towards trying to solve something. But there's also something very important about work-life balance that
Mhm.
I've recently kind of learning about, kind of, depending on who you ask, the hard way or like the easier way. And so I think more recently, like, I don't really typically work a 9 to 5. I am working a lot more than just a 9 to 5, depending on, when curiosity strikes, really. But there is a important benefit of firewalling, because if it's kind of like um, producing music or art or writing, and especially in science, you have to write. That's the most effective way of communicating your ideas and also being legit. um, you got to take a step. You got to take some time off, reset, because then when you go back, you might actually be like, "Oh, this makes no sense at all," or "Wait, no, this is a good start," and then that'll kind of fuel your creative aspect. And so I appreciate people that can work all the time, but, if you keep doing that, you can burn out. And so I try to maintain a healthy firewall, so that, at least I can come back and look at something with fresh eyes, whether it
Mhm.
be like a coding script, or a paper that we're working on,
Mhm.
so that it provides a different direction. So, to answer your question, I don't anymore. If people are interested, then I'll have a spiel that I lay out, but, I don't typically, go around saying, "Oh yeah, I do all this research at Truediagnostic," and things like that. But it is nice that my job is, I mean, I still think my job is pretty awesome, even when it's
Mhm.
stressful.
Yeah. I think another element, just to like kind of like emphasize something you were saying there, there you'll get more done with a good work-life balance. That's like I used to work like I used to not sleep for like 2 weeks and just work on a project, and that was just like I'm a little myopic, so like and obsessive, so like that's me. But with a better work-life balance, I actually get more done. With like let's say like I think there's like this myth where like the 40-hour work week was invented. I think the 40-hour work week was invented after like hundreds of people were shot when they were trying to like protest. You know, like
Yeah.
there was a time in our country where like hundreds of thousands of people would protest for the right to have Sundays off or Saturdays off or to have a lunch meal, which is why like I have friends who have like that structure at their work. They're like, "Oh, I'm not gonna have my lunch break." It's like so many people died for you to have this. Just take your lunch break.
Take it.
You'll work so much better when you come back.
Yeah.
And so like I don't know. Sometimes people always feel like they need to do more than 9 to 5 or less than 9 to 5 or whatever it is. But it's like it's about the depth. You know, if
Yep.
you I've been at places where you've done a week's worth of work in like in an hour, and but you have to sit there and pretend like you're working for 9 hours. Where if you just took a step back, maybe went for a walk, something would pop in your head, and you'd find something else to do, and you'd go even deeper on something else.
Yep.
That's a much better structure than this like manufacturing way of like I need to put 40 hours a week in or whatever. It's like have you moved the needle forward? Are you making these big contributions? I think that's more important. Um, and it's so I'm just saying like it's really great that you have that work life balance up because you're gonna do better work and you're gonna and given how important your work is, like that's good. It's a good thing. So I encourage this to anyone listening as well. If you do not have a good work life balance, just try to do like 10 minutes better a day or like 1 minute. Like things compound. If you do 1% different every day, you're like 3 times different by the end of the year. It's just like simple math right there. Um,
I mean,
the
it was something that, it takes time, trying to figure out. I mean, I had a terrible work-life balance, during my first 2, 3 years of grad school. But, you live and you learn, and, you kind of figure out what works for you. And don't get me wrong. I mean, there's like Saturdays and Sundays, where I'll just crank through like, scripting just because I'm just really focused and I want to crank through it. But then, you know, there's Saturdays and Sundays that, I don't want to,
mhm,
which is fine, as long as the work is being done.
yeah. All right. Have you because you're into music as well, have you found a song that you Pavlovianly triggered as you're transitioning off of work into in free time so that it's easier for you to leave and
Oh my
just like walk out without the bags?
No, I have not actually. Oh my god.
Well,
That would make a lot of sense though, right? Because if you have a song that just like, ultimately just turns you off into, like going home.
yeah. There's a there,
Yeah.
even breathing can do it too. That there are people that train themselves to breathe like four or five breaths a minute or something like that. And by doing it, it like allows them to reset and be much calmer when they're having critical things going on. It's like imagine a scenario where like you broke something, everyone's like yelling at you, and you take like those the if you've trained yourself right, you can be like the calmest possible. Like doctors do it a lot as well,
Yeah.
because like people die if they don't if they're not calm.
Right,
Yeah,
right, yeah.
I don't know if you're into The Office at all, but there's a period of time where I think Michael has like it's closing time. I'm not getting
Oh,
demonetized, but uh, I'm
yeah.
not monetized. But, and then like Stanley comes out and starts singing. It's like it's my favorite song now because I know I get to leave. Yeah, I don't know. Yeah, so at least in the fictional world, it definitely works, and Pavlov would say it would work as well. So if there were to be a song, and understand like you're gonna be listening to this every day when you want to clock out, so maybe like don't make it your favorite song or else you'll hate it. If you could find a song to be that Pavlov, what would it be? Like this is like totally not
Uh,
related. I'm just kind of curious.
see, so I don't have one, but, out of the songs that I could probably think of, "Get Back" by Ludacris probably would be one. 'Cause I think the essence of the song kind of, I think it captures how I feel when I wanna go home.
Yeah, it's like the criterion have definitely got to be like it's like quirky enough where like after the 100th time maybe you'll hate it, but like 50 more times you'll be like, oh hey, there's that thing that I like about it.
Yeah, yeah,
You know,
yeah, yeah.
so
Hey, that's
uh,
I've never actually considered that, but, I could see how that could work, and maybe I'll start doing that.
yeah. Well, if it works, let me know. I'll share the data with other people. Um,
Yeah.
are, have you? I think the biggest thing that I found when I'm talking to scientists, whomever it is
Mm.
the in the work life balance. It's the transition off. So I am curious if it's not Pavlov. What have you found other things that do work for you to transition off and like get your headspace so you can have a personal life?
Yeah, yeah. I think, and I will admit that, now in the, COVID, era, sometimes it's not, entirely possible, but what has helped me is to differentiate between office versus home.
Mhm,
During the COVID year, well, I mean, we're still in COVID, but, when the height of COVID was occurring and, everyone was socially distancing, that was really tough because, I always in a way, maybe the Pavlovian association that you were talking about was really affecting me then. Uh, I I could not separate, work from home because, I mean, I was working from home. And so one thing that I, do not take for granted anymore is working in the office, but then, limiting my work at home. Um,
mhm,
the only time that it gets a little blurred is during, Saturdays and Sundays, and even though I do work, some Saturdays and Sundays, I always treat it as like, I'm doing a little extra rather than saying I have to do it that way. And so,
mhm.
and even then, like, sometimes I'll come into work, on a Saturday or a Sunday just so I can have that different space, and so that mindset. And I think that it really comes into the play where once I sit at my desk at home, or sorry, at work, um, I can, there's something about it that I'm just like focused. I have my, dual screen monitor and everything. Like I just get into the rhythm of things and I get more work done in, let's say, 2 to 3 hours than I do at like 6 hours at home.
I think there's a Japanese saying where it says a man changes based on what room he's in.
Yeah,
And I
yeah.
think that's what you're saying. Yeah,
I would strongly agree with that Japanese saying.
yeah. So I have a couple more questions. Is it cool if we go a couple minutes over? I've been trying to
Yeah,
be
yeah.
on the okay.
Uh-huh.
Great, thank you. So
Perfect.
I have a question, and this is one of those where I was like, I don't know if I can ask this one well, but I have found as I've learned other languages, whether it's coding or not, that in learning the language, like a Spanish, for instance, like learning like the masculine, uh,
Mm.
like the L's and the laws and stuff, like and how that relates to the culture has impacted how, like if I'm speaking to someone who's like from that descent, it's like kind of an interesting thing to have that subtlety. And so I'm curious. I was looking and it says that a couple different languages, like Telugu, Telugu.
Yeah, so, Telugu, is my mother tongue, and I speak it, so I used to speak it very, fluently when I was younger. Now it's a little bit more broken just because of, not being able to practice it as much, but I do speak, whenever I'm back in, my grandparents' hometown.
Okay. Okay, great. So then I am curious, do the cultures that inherit that like every language is a culture. Like
Mhm.
it's like the bedrock, like it's one of the cornerstones. Um does that are you able are there elements in those languages that pull into your work or your critical or the way you address problems that you've noted? It's like I said, it's like a very weird esoteric question, but
Mm.
every now and again I wonder this, like how does language influence the way we address problems? Um so I was just wondering, in those languages, is there
So
anything in there that, yeah, influences you?
so that's, so, I guess, to answer it, not really, especially with,
Mm,
Telugu. And the reason why I say not really is because when I think of, sentence structure, I think of it from English. And when I speak Telugu, I have to translate it from English to Telugu. So in that respect, the reason why I bring that up is, a lot of, like, my work is, I guess, the first language that I think about in my head is English, and so that really translates over to being able to read uh, a lot of that, the scientific articles really doesn't affect that much. Um, however, eh, let's say in computer coding, they say that if the hardest language to learn in computer coding is the first language. And this is interesting because, like, like when I first learned Python, so Python was my first introductory computer language. There were a lot of cognates that really transferred over from, Python to R. And now Telugu being a Dravidian language versus English being more, I guess, an Anglo-Saxon Romance is it a Romance language?
I think it's a
It
Romance language.
Yeah, a Romance language. there are a lot more cognates between English and Spanish, and so like this, I think this is kind of translates over to how like individuals can really learn Spanish a little bit quicker once they know English versus let's say a Telugu speaking individual learning Spanish. And so I think in that respect, I think it's given me this, you know, I don't know if I'm answering your question, but I think that ability to, to learn one language, which was really difficult, but then that translates over to learning multiple different types of computer languages, and then allowing for a breadth of knowledge and different aspects. Maybe that's something that, I mean, taking Spanish during my high school, like that has helped with that. And then also kind of trying to identify cognates in Telugu from English, which actually I think there are a few. I mean, and also like Telugu, they just they do borrow a lot of English into some of the vernacular. So I don't know if that answers the question, but I guess
I
that's
think you did. It sounds like what I'm hearing is that there's a process for translating, whether it's a spoken language or it's a coding language, and that process is somewhat uniform in terms of like how you translate it forward and back into the language into the language that you use
yeah.
versus into the language that you're applying in a coding or when you're with your grandparents.
And because
Um,
and ultimately it's all based on logic. And
yeah.
in language, it's grammar, right? And then in computer language, it's computer coding. It's all essentially pseudocode and logic there too.
Yeah. All right. Well, thank you for taking a stab at that one. I wrote it down because I saw you had multiple languages, and it's like one of those that I always try to. I feel like there is a benefit in having multiple languages, which is why I'm always
Yeah,
somewhat sad that we're getting to the point where you have like the tele thing, like the babble fish in our
yeah,
ear that tells us everything. It's like you miss out on such a pillar of a group of people's uh, culture, and like then it's kind of like a piece of us, like all of humanity dies. But so it's something I'm curious about. So then before we transition to my rapid fire last few questions I ask, I am curious with all that you know about your subject, all that you give all these great talks, which I enjoyed. Um,
thank you.
is there anything that we did not capture? Is there anything that we left out that you really feel we should spend a little bit of time talking about? Like, because I hate to miss it.
I know. I think I think you allowed me to speak on a lot of the things I was passionate about, and this is one of the things is that there's so many things that are out there that maybe I'm forgetting too. But I think maybe really to restate that I guess when we start to think about the future of medicine, the epigenetic testing realm has been with DNA methylation and all that has been really an exciting field. And what we hope is that by creating a lot of these tools, that it really democratizes that ability to really know more about your personal health, seeing where you are and all that. And so I think that I mean, maybe to just restate, like, not only the work that we're doing at Truediagnostic, but, a lot of researchers and geroscientists and all that, creating these tools and then using these tools to see, what really works for you in terms of the aging process. Is, I think that's going to be the most exciting part. And I think now with a lot of the tools being built and being validated, I'm excited for the next however many years of geroscience and aging research.
Awesome. Um, then like a kind of a bonus one on that one. For anyone who's about to leave college, do you have any tips for them to find a place like to be involved in the way that you are?
Oh man,
Anything that yeah.
uh, I think be open minded to different opportunities, and then also if you're not good at something, don't let that be a detriment because you could always learn and teach and become better at that thing. And so, I mean, I credit a lot of my, I don't know if it's success, but like a lot of the positive, the trajectories to a lot of great mentors and people who were willing to be there to mentor me. And so you know if you have the opportunity to work at like some really great like really like top of the line company, versus a company where the individual is really the managing partners are willing to mentor you. Go for the mentorship, and I think that that is something that I don't know, not much. You can't really put a price on good mentorship.
It's the mentors are like the cartographers. If we're using the
Yes,
Lewis and Clark metaphor, they'll make sure you don't go down a quagmire. All right.
hopefully not.
Yeah. All right. So then my rapid fire question. So thank you again for letting us go a little longer.
Sure. Yeah.
Um, what is a problem you have that you love people's help with? If people are listening, you can say anything. What personal, professional? What's a problem you're having that you love help with?
Getting better at machine learning,
Okay.
or really applying the next level of machine learning.
What element of machine learning do you want help with?
For example, so we right now we have some deep learning elements that have been able to work, but like, how do we take longitudinal information and apply that as a as a feature to really improve on predicting the future state of that of, let's say, DNA methylation. So essentially
Hmm.
predicting where your pace of aging could be in, let's say, a year or so. And I think that there are methodologies there that can allow for that.
Okay. Sweet. Well, hopefully someone listening can help. And if not, I know some people I'm going to email after this. But anyways, what is a question you have? And if that's the question, that's cool. But what is a question you have that is unanswered? So like the one I always gave to give you space to think about it. The one I always gave is like if the if you took away the Big Bang, like what would be here? And so like it's kind
Mm,
of like the best way to think about it. The answer that I got was basically like you're describing like a where and a when, where you took away the where and when. So it's like I'm like defeating the purpose. Like where and when was defined in the Big Bang. So there would be no when or where without it.
I swear, ever since you asked me this question, I've been it's been ruminating in the back of my head. But yeah, what would happen if you if you ended aging? What
Mhm.
would this do to society? And what would it allow for, what would it do to, the consumption of resources? How to, overpopulation, um, the overconsumption of things that we need to live as humans. I think that's a maybe a societal and a question that, I don't know the answer to, and that would be whether we even solve it or not. I think sustainability is one of those things that I think about it a lot.
I think it comes probably like my like stab at the answer would probably like it'd be a culture shift. Like weirdly enough, it's not the fact that we're smart. It's not the fact that we speak. It's not the fact that we're fast or we can build things. It's like the invention of culture is what allowed us to go from like a bunch of people being eaten by lions to just wiping out every apex predator on the planet and being the dominant species. And that
Right.
was like us as a species has been around for like I think like quarter of a million years or something like that. But
Yeah.
it was only up until like the last 50 to 100,000 years where we were the dominant species. So like the last 150, like the majority of the time that we've been around, we were like nothing. But it was the invention of I was reading a great book on this. I can't see it or else I'd say it. But basically they were talking about how like culture was invented and like everything exploded. They call it the cultural explosion. So I'm thinking similarly there would have to be a giant cultural shift. Um,
Yeah,
that'd be like my stab.
no, it's a personal, it really is a personal, like, question that I have. Because yeah, like, 'cause, I work for a company that really wants to improve the human condition, and I truly do wanna, allow for individuals to be, to figure out ways to improve themselves and give that tool to them. But, I think, yeah, trying to figure out, to what extent should we do that, or is it more, creating a lifestyle that just is better, but maybe not extending human lifespan. I'm not sure, and so
Yeah.
that's why that question, I don't know. Maybe it'll be remain unanswered to me. Uh,
Yeah. Do you think you'll have the opportunity to explore that question in your lifetime? Or you think like this will be like maybe two generations away before they can really explore that concept?
I mean, I think what'll really answer that is, once we start to see individuals living longer, how does that,
Yeah.
affect, right? So it might be 2 generations after, and I'm not either for or against it. I just,
Yeah.
it's one
I
of
don't know,
those.
it's a curiosity.
Yeah, it's a question about, yeah, like, what would happen? Yeah.
Yeah. All right. And then last question is so for people who want to learn more about your field, are there books or resources in particular that you would recommend they check out? I think Google Scholar keywords definitely got that. But are there any capstone resources that you'd recommend people check out?
I think capstone resources. There are like, for example, for me, I follow a lot of professors and aging researchers online. So the likes of Dr. Morgan Levine, Dr. Stephen Horvath, Dr. Ricardo Marioni, Dr. Even Alex. Uh, I can't pronounce the last name, but there are a lot of individuals that I follow, and many of them have written books. And so I would highly recommend just at least going through their primary literature, but maybe just following them on Twitter and just seeing what they have to say about the field. Um, uh, and then people from the institutes like the Van Andel Institute, Buck Institute, places like that. Peter Verdeen is another individual that I'm thinking of. Gladyshev, Vladimir Gladyshev, I believe that's his first name. So yeah, so I think any of those people on Twitter first, and then try to see what books they have. I think that really helps.
I think the really cool thing about doing what you recommended is that once you're in the algorithm will help you go deeper. It's
Exactly.
like
Yeah.
you like Google pizza shops, and you're seeing pizza recommendations for a week. So you start Googling this type of stuff, and you'll start getting on the right algorithm for what you want to learn, which is
And
kind of a positive negative. So then, given the fact you're a musician, is there any music? I guess like as a fun send off, is there any music you'd recommend that we check out, or like is there a band, an album, a playlist that you'd recommend that we all listen to as we go about our day? And I'll link in the show notes so it's really easy for people to check them out.
Yeah, I don't know how many people would like it, but
They'll experience it regardless.
Exactly. You can experience. So there is a band that recently, I mean, I've been listening to them for the last about 2 years, really getting into them. Bring Me the Horizon, where they're from England. They used to be really screamo heavy metal kind of, but they've integrated a lot of like electronic EDM, like breakdowns and all that. And I don't know, it's a trip whenever I listen to them, because it's just blending all these different types of genres of music, which, I mean, they have truly a collaborative spirit, which I really appreciate as a scientist. And so, and when I hear it as a music lover, like oh man, it's great. You get like, I don't know, I would definitely at least check them out once.
Is there a particular song?
Oh, let's see. Teardrops. I think that's the one that comes to mind. Actually, I was listening to that this morning. So I listen to a lot of like heavy metal when I'm coding. It really helps.
Yeah, I mean, you want to get your heart pumping.
Yeah.
Like, it's basically like if you could work and walk, it's like the same effect.
Yeah, exactly, yeah.
And that was Dr. Run, where we talked about everything we mentioned in the beginning: methylation, biomarkers, genomics of axolotls, biogene clocks, why they're important, the future of the space, resources. I hope you guys enjoyed this. This is a really fun episode for me. We talked a bit before and after, and he's really a fascinating person. Just check the links in the show notes if you want to see something in particular. I hope you'll share, subscribe, tell everyone about this, because the more people who listen, the nicer it is. It's got a little bit of a validation. And so thank you, everybody. Remember to check us out at learnwithlowell.com, and I hope you all have a curious and fantastic week.